All right, it's 2 o'clock, so I think we should all get started. Welcome to this presentation in the Vitis Gen 2 webinar series. It's great to have people from all over the world joining us today. It's wonderful. So today's webinar is called A Low-Cost Core Genome Marker Platform that works well across the diverse Vitis genus. rhAmpSeq delivers 2,000 markers from 22,000 individuals, 91% transferable, covering 97% of the core grape genome. My name is Becky Weeps, and I am the Extension Support Specialist for the New York Statewide Viticulture Extension Program, as well as being part of the Extension and Outreach team for Vitis Gen 2. A few technical notes before we start. If you move your mouse to the bottom of the screen, you'll see a menu pop up. Click on the chat to open the chat bar, and you can use that to introduce yourself, make comments, and let me know if you can't hear something or see something that you're supposed to be seeing. Be sure that you change the chat settings to send to all panelists and attendees just so that everybody is communicating with each other. Also found on the bottom menu is a Q&A button. You can use this to ask questions. It'll help us to keep everything organized and asked in the same order. We'll stop briefly for questions after each speaker. We'll probably only take 1 or 2 at that time, and then at the end we'll try to get through as many as we can in the time that we have. Our panelists today are 3 fantastic Cornell scientists and members of the Vitis Gen 2 project. They are Chi Sun, Cheng Zhou, and Avi Karn. Chi is the Senior Research Associate of the Cornell Bioinformatics Service Unit and holds critical roles on the powdery mildew genetics and phenotyping and breeding teams. Cheng and Avi are postdoctoral associates at the Cornell Institute of Biotechnology and Cornell Agritech, respectively, and both of them are members of the Vitis Gen 2 genetics team. Thank you for joining us so much today, speakers, and everyone who is here to listen and learn. And with that, I'll hand it off to our first speaker, Chi.
Oh, thanks, Becky. So I'm going to share my screen.
Yep, that'd be great.
Okay. So Becky, can you make a screen shareable?
It should be. You're a panelist, so you should be able to go ahead and share it. Just go to the
I can't, sorry.
I will make sure that you can. 1 second. There you go.
Okay,
Is that better?
great. Yes, great,
Perfect.
thanks. Okay, can you see the screen now?
Yep, that looks great.
Okay, great, thanks. Thanks, Tim and Becky, for organizing the webinar. And thanks everyone for coming. So I'm Chi Sun, and today's webinar will be presented by 3 of us together. So I will start first, followed by Chen, and then by Avi. So 3 of us will present 3 different aspects of the rhAmpSeq platform. So the work we are going to present today is a work by the Vitis Gen 2 genetics team, and Lance Cato-Davidson, Jason Londo, 2 USDA scientists, has been leaders for the genetics team for the Vitis Gen 1 and the Vitis Gen 2. And the work we are going to present today are mostly done by 2 postdoc associates, and Chen and Avi, and they will be present their work in today's presentation. So briefly, rhAmpSeq is a unified genotyping platform across the Vitis genus, so it can be applied. So one single platform can be worked across the whole Vitis genus. And the current version of marker system we have can genotype 2,057 loci across the whole genome. And the rhAmpSeq genotyping platform can produce multi-allelic haplotype genotyping. And we'll show you that with multi-allelic haplotype genotype, we can get much better genotyping results compared with other platforms which give you the SNP, a biallelic SNP genotyping platform. So why do we need DNA markers? In a breeding program, breeder will start with multiple founder lines and through many generations of crossing and selection, and end up with small number of cultivar release. So during each cycle of generations, during each generation cycles, the breeder will do selections either through observed traits, like in class traditional breeding, or using model predictor trait. So for example, in marker-assisted selection breeding strategy, the breeder will select for individuals with desirable QTR alleles, or in the genomic selection breeding strategy, then breeders were using machine learning models for the selection. So in both mathematical simulation and real breeding practice, people have shown that using model predict trait can reduce the cycles and reduce the cost. So we know, so the bottom line is we need a good DNA marker technology for breeding to help with the selection. So here I show the history of the molecular markers technology have been used for grape breeding. So in 1990s, grape breeders started using the SSR markers for breeding practice. Then with the, so in 2011, so we started this USDA-funded project called Vitagen-1. In Vitagen-1, the genetic team started to explore new genomic technology for marker selection. So technology we have been looking at is called genotyping by sequencing, or short for GBS. It was a technology developed by USDA scientists at Berkeley at that time. So the reason we want to develop GBS is due to that the SSR markers being used by breeding, the issue is always the density for SSR markers is so low. We only have like 100 to 200 markers in the genetic map. So that caused the problems for that cause issues when we're doing genomic doing breeding, especially through many generations. So here I show the SSR mark comparison of the SSR mark and GBS mark. On the left is the SSR genetic map produced by Anne Fennell's group. So Anne is also a member of the genetic team of Vitagen, have been helping us with the marker development throughout all the years. On the right is you show the GBS map. You can see with GBS map, we can much high density compared with the traditional SSR map. So then we also look at another platform called rhAmpSeq. So the idea behind the GBS and rhAmpSeq map is that we want to have DNA markers. To get a DNA marker is through sequencing. But if we sequence the whole genome, the cost will be too high. So ideally, we don't have to sequence the whole genome. We only sequence part of the genome, especially target region of the genome. So the idea behind GBS is GBS is target the low methylation region of the genomic DNA. It's through the restriction enzyme. We know most of restriction enzyme are methylation sensitive. Then if we digest the DNA with a restriction enzyme followed by size selection, we can get rid of all the DNA which are highly methylated, and the remaining DNA are the low methylation, which mostly in gene-rich region. Through this selection, we can target to the sequence the gene-rich region of the genome DNA. So GBS works well for us for many years because we can have very high density markers and very highly accurate markers. But the problem with GBS, so I list a couple of issues we're having with GBS markers. Most of all is GBS system is very demanding for DNA quality. You have to use very high quality column purified DNA. So that in that case, if we have a very high scale genotyping project, DNA purification become a big cost. Also, analyze GBS data is not easy because, especially with the original GBS protocol, it is very high level missing data. Bioinformatics data analysis is very challenging when we work with GBS data. Also, the cost is not ideal because we are spending like almost more than $20 per sample per GBS to do GBS. So then we start looking at the amplicon-based technology. Amplicon-based instead of targeted low methylation region, we can design the PCR primers to target amplify certain region on the genome. And with the good thing with PCR is PCR use the Taq polymerase by use PCR is a pretty robust enzyme. It can work with relatively low quality DNA. And there's a low missing data. And we can achieve very, very high level multiplexing. So we started with rhAmpSeq in 2015, but the bottleneck for rhAmpSeq is if we want to if we want to pull many different markers together, many different set of PCR primers together in one PCR reaction, they start to interfere with each other. So we cannot multiplex in more than, say, 100 markers in per sequencing reaction. So that's become one of the major restrictions for us. So in 2018, on Lance, Jason, and I remember that time, Lance, Jason, we attend to the Plant Animal Genome meeting in San Diego. So we saw an exhibit by the company called IDT. They present a new technology called rhAmpSeq. So at that time, IDT was promoting the rhAmpSeq as a technology for SNP genotyping. So we saw the opportunity to actually modify the rhAmpSeq technology to use for haplotype genotyping. So Chen and Avi will tell you more about the work on developing this rhAmpSeq genotyping markers today. Before I switch to Chen, I just want to say a few words about what's a good marker system. What's the criteria for a good marker system? So we think a good marker system must be transferable, must be accurate, and a low cost. I don't need to explain that markers need to be accurate and low cost. They're obvious, but I want to say a few words about what's being transferable. So when I say transfer, means a markers need to be transferable across generations in breeding cycles and has to be transferred across breeding programs. So ideally, we just have one single universal marker system can work across all the breeding program, across all the generations. So in order to achieve the transferability of the marker, one very important thing is the marker type. So when we look at SNP-based marker types, so what we have seen is a SNP marker tend to be biallelic. That means for every marker, for genotype, you can only have two states, major allele or minor allele. With only two states, if we want to track a desirable QTL allele, then through several generations, especially when we introduce a new parent from different genetic background, very easily we lost track of the desirable allele we want to track because we only have two possibilities, major and minor, right? So then we have been, however, with haplotype alleles, that's very different. With haplotype alleles, it's multi-allelic. What we have seen is by genotyping the germplasm data from USDA gene bank, what we have seen, each marker could easily have dozens or even hundreds of states. So when we track a desirable QT allele, only one of these 100 states can be associated with one particular desirable QT allele. So that's much easier through generation. We can just track this particular allele throughout the generation. So that makes the marker much easier to track, much easier to do selections. So we would prefer to have a haplotype-based multi-allelic markers. Then we know the genome structure could affect the genotyping accuracy. So preferably, the markers we selected should be regions of low genome structure variation. So if we have deletion in the genome, if we have a parent with deletion in the genome, that could lead to missing data in the genotyping results. Or if we have duplication or translocation on the genome, that could cause alignment errors and could lead to genotyping errors. So we would preferably to design markers in structure stable and low genomic structure variation. So I will stop here and then we'll switch to Chen. We'll tell you more about the rhAmpSeq marker design.
Thank you so much, Chi. Does anybody have any questions before we move on to Cheng? There's nothing that's come into the question and answer box, but feel free to submit something now if you'd like to ask Chi a particular question before we move on. Nope. All right, well then go ahead, Cheng.
Sorry, I muted myself. This is Chen. I will continue to introduce the marker development based on the Vitis core genome and the primer design of this rhAmpSeq genotyping panel. So first, I would like to emphasize why we need a universal marker system for the Vitis genus. As we well know that grape is domesticated in Europe. Therefore, when this domesticated cultivar are introduced to US or other places, they are susceptible to many local diseases. While the local wild Vitis species have co-evolved with major disease for a long time. Therefore, the wild resistant species can be used to help cultivar to adapt to their local environment. So for example, in US, different states has been using very distinct genetic background to improve the commercial cultivars. For example, in Minnesota, they tend to use Rhiparia robustus to improve the wild wine table breeding. And in New York, we want to use Scenario and Labrusca. So in different states, we are using different wild species to improve the cultivars, and also we have very diverse breeding targets like table grape, raisin, or wine grape. So, but why it is challenging to develop a universal marker system for the Vitis genus is because the Vitis genus is highly diverged. For example, we have known that there are at least 28 species in North America, 30 species in Asia, and 1 species in Europe. And they are diverged about 28 million years ago. So at this genetic distance, only 2% of the SNP markers are transferable based on our previous study. So how can we increase this 2% transferable rate to an acceptable rate? Because the Vitis genus is so divergent, one reference genome is not enough to represent the whole genus. So the first thing we have done is to de novo more reference genome in the Vitis genus. So as a group effort, we assembled 2 wild species, 4 hybrid, and 3 commercial cultivars, which are representative of North America germplasm, wine grape and table grape. Most of them are assembled using 10x genomics, and some of them are assembled by PacBio or Illumina. The scaffold ranges from almost 300 kilo base pair to 2 mega base pair. For all 9 genomes, more than 90% of the basal genes were represented in full length, indicating a good coverage of the gene space. So the next step, after we get a set of high quality genome, we construct the Vitis core genome. So the first thing is to align these 9 assemblies to the reference genome PN4024 individually. Here, we are not only recurring the homology between the query and the reference genome, we also consider the order of the alignments. So the order of the alignments must be in linear. For example, the alignment in black were not included in the genome alignment because the order of the alignment is not collinear with the environment. So next, after we get individual pairwise genome alignment, we project all the alignment to PN4024. So for one region on the reference genome, if it is covered by 9 assembly, we define it as core genome. If it is not, we call it dispensable genome. So here is an example of the coverage chromosome 2 across the 9 genomes. Each block indicates a collinear genome alignment. On average, only 37 of the reference genome was collinear with wild species. But in cultivars, this number raised up to 54%. So in total, only 10% of the reference genome are also present in all 9 genome assemblies. The total length of the core genome So yeah, the total length of the core genome decay exponentially as more assembly included in the core genome. So do we have enough genome yet? By fitting this exponential decay, we inferred that the core genome size in Vitis is predicted to be plateaued at 39 megabase pair with 17 assemblies. So although we do not have 17 assemblies to construct this core genome, but with our 9 assembly, our core genome is approaching to the plateau. So yeah, in our core genome, it's 42 megabase pair, but in this fitted model, the predicted core genome is 39.8 megabase pair. So it's very close to the plateau. So certain genome, we also find that certain genome features are enriched in the core genome. So here is a circles plot. The 6th track is the density of the core genome, and the 1st track to the 5th track are some important genome features like gene density, density of transposable element, and a different kind of transposable element density. So we found that the core genome is enriched in genes, but is depleted in transposable element, particularly the most redundant transposable element family, gypsy. So here is what we have done to construct the core genome, core genome for the Vitis genus. So next, because this core genome is constructed by line genome, so we want to evaluate polymorphism using more individuals. So here we evaluate polymorphism using 50 individuals from wild species and cultivars. Each line here indicates a accession that line to the reference genome. The dot indicates one variance. So we do not want the region to be too conserved. In this case, in the left-hand side, there are very few informative sites there. But when this region is too polymorphic, there will be lots of error there and probably misalignment. So we only choose those regions with moderate polymorphism. So after we collected the core genome and evaluate the polymorphism, we pick up candidate region for this design. So the criteria we used: first, the candidate region must lies in the core genome, and then we require a moderate level of polymorphism, and we want it evenly distributed across the genome, and also we add some more candidate gene region. So next, I'm going to introduce a little bit about this platform we used. So it's commercialized by IDT. So different from the traditional rhAmpSeq, the rhAmpSeq adding an RNA base and a blocking moiety after the primer. So only the primer and the target is a perfect match, and RNase H2 will cleave the blocker. So this PCR can only be extended when the primer and the target is a perfect match. So in this way, the primer dimer and other undesired amplification products are reduced. So at this time in our design, we have 2,057 loci, and in one lane, we can pull 96 samples per plate, and we pulled 48 plates per one lane. But this design can be expanded to 5,000 sites, and using this dual indexing barcode, we can easily pull up to 72 to 96 plates per lane. After we handed this candidate regions to IDT, they will do the primer design for us. So sometimes there are regions they cannot design. So for example, here we gave them 2020 sites. They are able to design for 99.5% of the sites. And also they need to consider the compatibility among those primers, because we need to pool all those primer in one tube. So after this pooling verification, they find that about 98% of the primers can be pooled into one tube. So in general, the design rate and the pool rate is pretty high. So in most time, if you handed them a region, a primer can be designed for this region. So here is a histogram of the assay size of these 2057 markers. So in most of time, the assay size is bigger than 200 base pair. And we did a pair-ended sequencing from each end, and there are some overlap of the reads. And instead of I want to also emphasize that when you get a result, you get the 2 haplotype for each sample. So in this way, you do not need any imputation or phasing. It will give you all 2 haplotypes when the red depth is enough. Yeah, there are some cases the red depth is not enough to find 2 haplotypes, but Abby will introduce that in the next slides. Yeah, that's all my slides, and I would like to take questions.
Great, thank you so much, Cheng. I did also want to note before we move on to Avi's presentation that I am recording this webinar. I know that with the webinar platform, people who are watching can't record it themselves, so I am recording it, and it will be circulated to everybody who was registered for today's presentation. So you'll be able to go back and kind of look over everything. And with that, I guess we will start with Avi's section.
Okay. Hi, everyone. My name is Avi Karn, and I'm hoping everybody is doing well during this time. During this part of the webinar, I'm going to talk about the validation effort that we did for rhAmpSeq core genome markers. So for the 2000 rhAmpSeq, excuse me, rhAmpSeq marker panel, we evaluated the marker set in 4 grapevine families, representing a wide range of genetic diversity in US breeding program. We can see, so we can see the genetic diversity and the genetic and structural diversity in the cluster plot here. So these 4 families came from 4 different breeders across the United States. For example, F2 population came from Ann Fennell's breeding program. Similarly, a modified backcross population came from Dr. Craig Ledbetter's breeding program. And the one interspecies F1 population was from the Bruce Rice's breeding program, and the 4th population, which was an intra-species F1 population that came from Matt Clark's breeding program. So I'm gonna just be mentioning each population by the breeder's last name. For example, I'll be saying Fennell or Rice or Clark or Ledbetter, just for easiness. So once we got the pair and sequencing data of rhAmpSeq marker from all these population, we call the haplotypes using an in-house script or in-house pipeline to call the haplotypes. So the first thing we wanted to look at, how does the PCR implication and the sequencing bias, how much bias is there in the read depths per sample and per reads per marker. We found the markers and the reads depth were highly uniform. given that we use two different sequencing platform. Also, we wanted to see how well the marker, how many out of 2,000 sets return data across all these four diverse families. So we found, our analysis suggested that we have over 91% of the marker return data across all these four families. And we also wanted to look at how the alleles look like in this four with the structural different population with a different structure type. The alleles, most of the markers have alleles segregated in a Mendelian fashion. Also, each rhAmpSeq marker had an average of 5.7 alleles across the genotype parental accessions. The next thing we did was as a part of the quality control analysis, we wanted to build genetic maps for these four. These were parental genetic maps for these four families. We use a LepMap3 and LepMap3 software. It's an open source software to make the genetic maps. So in the left the scatter plot, we see that about 90% of the markers return enough data, but about over 60% of the markers ended up in the linkage map. We also found there's a strong correlation with the genetic and physical position that are you can see in the X and Y axis. Also the genetic maps depicted the structural, the variation that exists in the grape germplasm. We also saw that this on the right hand side is a circles plot, just to show and give you an overview of the genetic map, the markers that ended up on genetic map, and out of the 2,000 markers. So our circle A depicts the 19 chromosomes, and B depicts the marker density, 2,000 across the genome. The C circle is the marker, are the markers on the consensus map. So our approach was another approach was to make a consensus map following the joint linkage mapping analysis. So we were able to add over 83% of the markers on the genetic map, which we can see in the green dots. And then the black dots represent the markers that were distorted, and the white dots, that's hard to see, but these markers represent non-informative. And there are few red dots that represent the markers that were mismapped that ended up a different linkage group than they were from their original chromosome. Like she mentioned about the marker transferability, it's been a big issue of, when before AMP-seq or rhAmpSeq, with the GBS marker, we had a big issue with the marker transferability, meaning that when we had a QTL, we perform a QTL analysis with the GBS marker, well, we would have a different markers in different families that were linked to the trait. So to evaluate the marker transferability, we picked a flower sex trait for this interspecific F1 hybrid and the F2 population. And we performed a genome-wide association analysis to see how the marker, what markers are significant. So in the analysis, after the bond-forming correction, we found the same marker to be linked that they linked to the QTL, explaining the highest phenotypic variation. Like I saw Tim just posted the link to the paper. So this whole analysis has been posted online. I mean, it's been published last January, and it's open access. You can read more about it. The next thing we did was we screened. We used the same marker panel, the 2000 rhAmpSeq marker, in screening the rotundifolia genus species. And we did a similar GWAS analysis on a flower structure, and we found the same marker to be linked to the flower for the flower type. And recently, we also used the same marker platform the rhAmpSeq marker platform to genotype over 21,000 grapevines that belong to vinifera, muscadine, vitiaceae, wild and hybrid grapes. So with these markers has successfully elucidated the genetic structure of these different species quite well. And also we looked at how much missing data, meaning what proportion of the marker fail to return data across all these species. So vinifera had the lowest missing data, whereas the Ampleopsis had really high missing data. So another thing we did was with all these good quality genetic maps, we have been performing a QTL analysis that have very consistent QTL profiles. And with that, we have been also able, with the 4 haplotypes, with minimum 4 haplotypes per marker, we have been able to identify, accurately identify recombination events, which is useful for the fine mapping analysis. In the future work, I have also done some preliminary analysis on genomic prediction. With the rhAmpSeq, I have done some genomic prediction with the rhAmpSeq marker and the GBS marker, and found that the haplotype alleles tend to have more prediction accuracy in comparison to the GBS marker. So in future, I'm hoping to use this marker platform for the prediction and selection, genomic prediction and selection analysis as well. And we are also hoping to perform marker-assisted selection using their core genome markers for different powdery mildew-resistant loci, downy mildew-resistant loci, seedlessness, flower sex, and so on. So to get started using the grape genome, our grape genome rhAmpSeq core panel. So this is just an announcement that I would like to make. We process by plate of 96 samples, and one has 2 options. First option is to ship the good quality DNA that is PCR competent, preferably from Kaizen columns or similar. Per 96 well plate, we invoice about $1,046.40. Second option is to ship the leaf tissue to Intertech Sweden at the following the email address. They all provide the protocols, so one can isolate the DNA at about $200 per plate for public researcher, and then they ship the DNA for rhAmpSeq. And again, at $1,046 per plate. And for the price, we return rhAmpSeq raw reads and return the alleles to you. And if anybody is interested, I would suggest contacting Dr. Lance Kettle-Davidson for more information. With that, I would like to thank all the attendees, also our funding resources and the Vitis team, especially our extension team members, Tim Martinson and Rebecca Waipiz. And thank you. We'll take any questions.
Great. So we do have a couple questions that have come in, and there's a couple that you guys have answered just by typing, and it would be great if we could just read those out so they're part of the recording. So Bruce asked if you've tried the 96 well plate, or the 5,000 marker 96 plate max with good results. And Lance, I'm gonna allow you to talk if you'd like to contribute what you were typing in.
Okay. Yeah, can you hear me okay?
Yeah, you sound great.
Okay, great. So great question, Bruce. So we have now done up to 2,057 markers per multiplex. So the original set was 2,000 targeting the core genome. And then we had markers that we had previously been using for marker-assisted selection. And so we went back to IDT and asked them to use their computational pipeline to add more markers in. And of the markers we wanted to add in, not all of them worked, but I would say about 95% they were able to port over. So it may not be the same primers that were originally used, but it was the same region. So we may have to shift the primers around a little bit to get them to work in the multiplex and at these annealing temperatures, et cetera. So I think there's no theoretical upper limit on how many amplicons you can multiplex in rhAmpSeq, or maybe even in AmpSeq, I don't know. But for us, we're pretty good. I think at 2,000, 2,500, somewhere in there is probably all that we'll, in the near future, have in our multiplex. Now, regarding going from our standard 48 plates per sequencing lane to something like 96 plates, that's definitely doable. We currently have 72 plates worth of barcodes and have run experiments with, I don't know, probably 64 plates in a single lane of sequencing. And it's just a trade-off between the cost that you put into sequencing per sample and the depth, the sequencing depth that you get out, you know. And so you want to make sure that when you add more markers and more samples, it multiplies by each other, and you lose sequencing depth per locus with that. And so you want to make sure you're getting enough sequencing depth for your application. If we go to 72 plates, we're only spending about 67 cents per sample on sequencing. If we go all the way up to 96 plates, we're saving about 17 cents per sample, spending about 50 cents per sample. So it gets to be less and less a benefit to multiplex higher and higher. And I'll also comment, Daniela sent a really good question about whether we can redetect fully informative SSR markers. And I think the answer kind of fits with the portability from our marker-assisted selection markers over into this 2000 plex. And that just depends on whether, as Chi pointed out, the bioinformatics at IDT are really strong. And if they say something is going to work, we find almost 100% of the time it works. So there will certainly be some SSR sites that will not port over into rhAmpSeq and rhAmpSeq, but I think a lot of them will, and it's just a bioinformatic process to get those to work.
Yeah, so in the same question, I also ask whether we only prefer the SNP variation. I guess that's the question is. No, we don't. So we basically, any polymorphism can be scored in this genotyping platform. It could be SNPs or could be deletion or insertions. As long as the variation within this 300 base pair region, we can detect it.
Great.
Yes.
Go ahead, Chi.
Also, there's a question about how are these markers coded and treated in mapping programs like SNP or what? Okay, so in our genotyping platform, we produce haplotype genotyping results. So in the raw output, it's coded a numeric allele. So it could be allele 1/2, 3/4. So we can have hundreds of alleles, numeric allele IDs for every. So the primary output from genotyping pipeline is like SSR. We give you numeric allele ID. Then in all how they treat it in the mapping program. So it depends what kind of program we're using. We have found some mapping programs like the LabMap program we have been using require ACGT, the nucleotide alleles. So what we have been doing, we provide pipelines to can pipe into either the fake ACGT alleles. Also, we can pipe into we also have a pipeline to convert to the 4-way cross RQTL 4-way cross coding system. So we have different so briefly, it's basically the pipeline produces a numeric allele ID, and which can be converted into any coding system of the mapping program. Could be either fake ACGT alleles, or could be numeric alleles, or 4-way or 2-way cross coding as in RQTL.
Great, and there's one other question that came in on the question and answer chat. If one of you would like to answer that, would that price be the same or similar for a similar number of markers for a different species?
I guess for Lance?
Sure. That's a loaded question. The pricing is all negotiated with IDT, and their list pricing is quite high because one of their main markets is the medical community that can afford to pay more than an agricultural scientist typically does. So we've worked very, very closely with IDT to share with them what our price points are and what we can do to help drive that cost down. In getting it up and running in a new species, it's the upfront cost of buying the primers that is really probably going to be the limit to a lot of people. You know, for us to have 2,000 markers, the list price on that would have been about $80,000 up front. Now, once you have them, okay, the cost per sample is really quite low. Their list price on reagent is about $16 a sample, but they're very willing to negotiate and drive that price much, much lower, which, you know, I think Grape is doing about as good at pricing as people will be able to do in other crops from what I've seen, because we've been trying to work with IDT to get other crops up and running. But you know, that $10 price point where we are right now, $10 or $11 per sample, is probably a good goal for people getting up and running. And that price includes the cost of the primers, but we bore that cost up front and then pay it over time. Yeah, and so the thing that we're really recommending to people is think about not just the crop that you're working in, but the related taxa as well. So if you're working in rosaceous crops, we've shown that this strategy can bridge taxa that are about 30 million years diverged. And so we think that a core genome strategy around a set of species or whatever taxa about that far diverged should be a way to leverage more samples and get the costs down.
So I want to say IDT is not only company provide multiplex amplicon sequencing solution. The other companies provide similar service too. So for example, there's a company called AgriPlex, and they also provide high multiplex PCR solution. So you don't have to go with IDT. You can look into, so basically for us is we have designed the regions we want design markers for, then we contract IDT to design primers for us for genotyping. But you can also look into other companies, right? American AgriPlex, I think they also other company provides a amplicon-based solution. You can look into other companies to see whether you can get a better price point.
Great. And then we also had a question come in on the chat. Is the technology applicable to polyploids?
I think I can answer that. The answer is yes, but I didn't mention that when you design primers, you must be very careful, or you must work on multiple genome, or if you, as a polyploid, you must have the assembly for each subgenome. In this way, you have to avoid mismatches in the primer, because when I design, I avoid mismatches in the primer. If the primer bite into different subgenome with mismatches, so the amplification may fail for some specific subgenome. So that's the thing you want to pay attention to. Otherwise, if the primer works for each subgenome, the technology should work, and you should also consider about the sequencing depth, because you have a polyploid, probably you need to sequence more. Yeah, that's my thoughts. Yeah.
Yeah. So my take is with polyploid genomes, actually SNP markers is very difficult to work with if you have polyploid genome because SNP markers only have 2 alleles. And in our case, we see many more alleles. So we actually, for any markers, we can get like dozens of alleles. So it's more likely with haplotype markers, it's more likely to find polymorphism between the subgenomes. So in that case, it will work. So I would say it will work much better than SNP markers if you're working with polyploid genomes.
And there is one question in the question and answer bar still. Do I need to dilute the DNA samples to a certain concentration?
Lance, do you know?
Yeah, I can deal with that. We've dealt with a lot of. In fact, I just sent an email during this webinar to somebody who had asked that question, and so let me pull it up. But basically, we've dealt with a lot of DNA from different sources, and we report on some of the effects of DNA quality in the Nature Communications paper. One group, so we get beautiful results if we have good DNA. We had one group that did CTAB DNA isolations. They were able to get SSR data with that DNA, but it was several years old, and it did not work well at all with rhAmpSeq. So it's kind of a cautionary tale that not to use poor quality DNA. But Linda, who's the technician who runs the library preparations for rhAmpSeq, she uses a robot and likes to have at least 10 microliters of volume. And her guidance is if your concentration is lower than 2 nanograms per microliter, which is pretty low concentration, she likes to work with 15 to 20 microliters of DNA. So it doesn't take a lot of DNA. It's a PCR-based technology.
Yeah, this is Tim Martinson. I'd like to chime in here with a couple of questions and maybe some comments. First one for Avi. You described the breeders that originated the 4 populations that you use, but could you tell me a little bit about where their programs are located for the audience and what their programs address, because they are very different breeding programs.
So I'll start with Bruce Reich's breeding program, which is located in Geneva, New York. And Matt Clark's breeding program is located at the University of Minnesota. I believe it's Twin Cities. If I'm correct, somebody correct me if I'm wrong. And Craig Ledbetter's breeding program is Parlier, California, and Ann Fell's breeding program in South Dakota State University, in Brookings, South Dakota.
Right. So this is a lot of diversity in the breeding programs, including table grapes, very cold, hardy cultivars, and then things that are more suited to our climate in New York and in the East. So I have a kind of a broader question for the group. So since you've got this new technology, and maybe Lance can address this, what has it allowed the VitisGen project to do? What things have you discovered as a result of having this mapping technology?
I'd love to hear Avi and Cheng's response to this. Maybe can I set them up first and then follow?
Sure.
Okay. Because they're doing, day to day, they're doing the analyses and they're really doing a phenomenal job. And so, Avi, maybe you want to start off by talking about some of the traits that you've been able to look at in a big picture, and whether the markers that you're finding that work in one population work in another population.
Yeah, so one of the, when I started, one thing that I really noticed working with the GBS versus the rhAmpSeq marker. So the first approach has been to look at how much the amount of data we have, the quality of data. Also, there's much less missing data, which does not require imputation. So that was one thing. And with the building genetic maps, since we have a haplotype, which has much more information than a binary SNP marker, it's much easier to phase the marker and estimate correct recombination frequency, which definitely makes things much easier during the genetic map construction. So when looking at the GBS genetic map, the maps are super inflated, and with a lot of erroneous SNP calls. But when I look at the rhAmpSeq marker data, genetic map, the map, they are below, most of them are below 150 centimorgans, even less. So that's the quality definitely is much higher with the rhAmpSeq core genome markers. And another thing I have noticed with QTL mapping, we have been able to capture enough variation per marker with the 2000 core genome set. Also, and since these markers were targeted to the gene-rich region, we are able to track strong candidate genes, which was before, when we are looking at the GBS marker, it's hard to find the candidate gene after the QTL analysis. And fourth thing I have noticed with the correlation with let's say we have all this core genome marker, and when we look at the physical position and the genetic position, there's a strong correlation between the genetic and physical position, which was a big issue with the GBS map, because markers, if I make a correlation plot, not all the time, but in several incidents, I would see markers with the shuffling, so it's hard to get that physical coordinate to identify candidate gene regions, So also, like I mentioned about the genomic selection and prediction work that I'm just initially basically touched on. I have just used simple Bayesian regression to do some prediction. With the GBS marker set, the prediction accuracy was much lower with the moderate heritability trait. But with the rhAmpSeq marker, I have a much higher, I would say about 70% prediction accuracy with only let's say 90 samples in a training set. Yeah, those are the things I can remember right off my head, talking about the comparing the GBS and the rhAmpSeq markers. Yeah.
Thank you, Avi.
Yeah, sure.
So more specificity that allows you to really hone in on those genotype, phenotype associations more accurately and predict candidate genes.
Yeah, the specificity is like when you have a primer, the RNA, for example, that RNA. Small RNA sequence. I mean, no, it's not a small RNA sequence. It's about 4 nucleotide RNA sequence. That is, it helps in targeting the target loci. I would say the loci. So we are not misamplifying another part of the genome. So we are not we are not having issue with paralogs or any duplicated reason. But and we are also not seeing PCR biasness, or we have low competing markers. And that helps. That also helps in having a uniform quality data.
I'll just chime in real quick here, and because I see we're at about an hour, and I don't know if we have extra time, but for me, the most fun and exciting story for this came last summer when we had a visiting scientist from Germany, Dr. Daniel Schindler, here working on the REN3 and REN9 locus. And we realized that we had a lot of germplasm for REN3, REN9 that we had already genotyped. And overnight, we were able to screen about 2,500 seedlings, since we had run them all with the same marker platform, and identify recombinants at those loci, and provide really useful information, I think, to his project. So, and the next step here is we've now genotyped all of the USDA collections with this marker platform, and are very interested in genotyping collections around the world. So as we identify alleles that are linked with genes, we can very quickly point breeders to germplasm around the world that has that trait of interest that they can turn to and use in their breeding programs.
There's also a very important question from the chat, and what's the turnaround time? Lance, do you want to talk about it?
Yeah, I can try to estimate it. So right now with DNA isolation in Sweden, which is what we've decided to do to outsource it, basically the breeders ship internationally to Sweden. You know, that takes about a week basically to get the leaf samples there. They usually within the work week can turn around thousands of DNA samples and ship them back to Cornell. And then the turnaround time, you know, this is a university service facility, so it depends on what other samples are coming through at the time, but usually it's weeks, and then the data analysis, depending on the number of samples that we're bringing through at a time, because we often hold samples up in order to have cost efficiency and time efficiency. You know, it may be a couple weeks, it may be a month or two. If there's urgency, then we push it faster. But the computational time, when we did the full bill on 20,000 samples, that was what, about a week's worth of computation. Anyway, hopefully that gives you an idea. The throughput could be faster. It could probably be about a 2-week turnaround time if you really were wanting to turn the crank fast. But for us, we're usually looking at 1 to 2 months.
All right. Thank you everybody so much for attending. This webinar has been recorded, and I'm going to circulate a link to everybody that was registered for today's webinar. It's also going to be posted on the website. And then I'll also be circulating to those of you who attended today a brief survey asking for feedback about today's webinar. It only takes a couple of minutes to fill out, and we really appreciate it. It helps us in all of our future webinar planning. Thank you again to all of our speakers today and to Dr. Cadel Davidson. And thank you all for joining us so much. Yeah, thank you.